All right, good morning. Today's topic is going to be the cell membrane and transport in and out of the cell. Mostly when we talk about transport, we talk about osmosis if it is water, diffusion if it's small and going with the concentration gradient, and active transport if it's a larger molecule or it's going against concentration gradient. When we talk about getting things in and out of the cell, we got to talk about the cell membrane. You are almost always going to have to draw or label or on a multiple choice question, identify the parts of the cell membrane. So make sure that in your notebook that you draw and annotate this cellular membrane. The big ones here are always the integral proteins. The integral proteins are the ones that go all the way through. When we talk about cell transport, these integral proteins are the ones that are going to bring things from outside to inside the cell or vice versa, because they do breach across the cell membrane. Peripheral proteins, these are ones on the surface of the cell membrane. They're going to be used for cell-to-cell communication or for accepting hormones or other kinds of cell signaling. Cholesterol is there. This provides support and flexibility within your cell. We have our glycoproteins and our carbohydrates. They all have functions in the cell. Anytime that you see a movie linked on the screen, you do need to watch that movie. These movies are designed to give you greater in-depth understanding of the topics that I'm talking about. IB likes to give you the electron microscope view of the different items that you're looking at. And so this is an electron microscope view of your cell membrane. Now note that it doesn't have a great detail. So you're going to see your cell membrane and the intercellular space, but not all of those integral proteins and stuff. The properties of the phospholipids in the cell, and let me just move myself to another corner. I wish I could get rid of that, but I can't. Okay, so a phospholipid is composed of a hydrophilic end made of a phosphate head, which is polar. Hydrophilic, it loves water. So this is going to be on the outside or the inside of the cell. It's going to interact with water. This is different than the hydrophobic end, which is a fatty acid chain. It's nonpolar. The hydrophobic ends line themselves up towards the center of the cell membrane because they don't like being around water. This nonpolar middle and polar exterior make it effective in preventing molecules from entering and exiting the cell that the cell doesn't want. This is why it's a barrier. It basically, polar and nonpolar molecules run into roadblocks. And that's why we have these integral proteins that are going to be doorways or gates into and out of the cell. We have membrane proteins, and they're going to have a bunch of different functions. So integral proteins, the majority of the protein is found between the phospholipid molecules. They interact with the cytoplasm on one side and the inside of the cell on the other, or the outside of the cell. This is going to be that gate, like I said. Peripheral proteins are found mostly on the outside. They interact with phosphate heads and are not permanently associated with the membrane. So they may be found on the cell membrane when that cell needs to accept hormones, but when that cell's not accepting hormones, they disassociate or they get dissolved. Glycoproteins is a carbohydrate that is attached to a protein. And then cholesterol is a large molecule positioned between the fatty acid tail of the phospholipids, reduces fluidity and permeability of the cell. So the more cholesterol you have in the cell membrane, the less things can get in and out. Membranes with a lot of cholesterol are very stable, not very permeable. If you have way too much cholesterol, then your cell membrane is kind of like a shell of an egg, which is not a great thing. So you do need some cholesterol in your diet, just not too much. It allows the cell to withstand changes in temperature. Again, here is another picture of your cell membrane. You need to be able to draw this and label it and understand what all of these different proteins and parts of this cell membrane are doing. We do have membrane proteins, and they have a bunch of different functions. So hormone binding sites, we have hormones that are traveling through your blood, and they're only going to interact with certain cells. So estrogen, only interacts with certain cells of the body. We don't need estrogen affecting your heart. And so the heart cells aren't going to have a receptor for estrogen. Immobilized enzymes. These are going to allow a sequence of reactions to occur. Think about on your mitochondria or on your chloroplast. This is what's going to allow respiration or photosynthesis to occur. Remember, the chloroplast and the mitochondria, we think were once independent cells. They have a membrane that have all of the components that the membrane of your cell have. So that endosymbiotic theory where the chloroplast and the mitochondria were absorbed into another cell and then became like a symbiotic relationship, we're going to see these immobilized enzymes on the membrane of those two organelles. Cell adhesion, so integral proteins bind to specific proteins on neighboring cells. They also bind to the extracellular matrix. The extracellular matrix is like a basement membrane that all of your cells will sit on. Cell-to-cell communication, direct contact between cells, signals as hormones and neurotransmitters, could be triggers to divide. If two cells aren't touching, then you're going to have a trigger to go through mitosis to get new cells until the cells touch again. Channels for passive transport and active transport. Extracellular components. These are things that are non-living parts of the cell that are outside the cell membrane. The two big ones here are cellulose, which is found in plants, and a basement membrane, which is found in animals. In cellulose, this extracellular component is the cell wall. It's going to provide support and function and shape of the cell. It is going to prevent that cell from bursting under osmotic pressure, and it's going to prevent pathogens from getting into that cell. It's made of carbohydrates and can act as an energy storage site. So if the cell is dying, right, it doesn't have enough sugar it needs for its kind of processes, it will actually start breaking down its cell walls. This is not sustainable. It's a last-ditch effort for these cells to stay alive. Your basement membrane is made of collagen and glycoproteins. It's found in animal cells, and it basically prevents stuff from seeping out of organs and acts as support for the organs. So think about your stomach that has all of this hydrochloric acid in it, that pH of 2. You want to maintain that in the stomach, and so the stomach is surrounded by this basement membrane. And so the cells sit on top of the basement membrane, and there might be small gaps between those cells. This basement membrane prevents any hydrochloric acid from seeping between the cell membranes through those gaps and then out into the body cavity. Diffusion is our next topic. Diffusion is the movement of materials with no energy required from areas of high concentration to areas of low concentration. So my example of this is, imagine you are all sitting back in my classroom and there's 28 of you, and I shove you in the corner of my room and I walk away. Are you going to stay in that corner of my room? Probably not, because we all have our own personal space that we like to be in, and we need that personal bubble. And so we're going to spread out in that room until we all have our personal bubble. And we're going to keep moving in that room to maintain that bubble. If someone takes a step towards you, you take a step back to keep that bubble. This is diffusion. We're going to diffuse or move around the room until we are all equally spaced in that room. It doesn't take energy. I didn't have to tell people to move away from that corner. They just naturally did it because they want their bubble. When we talk about the cell and the cell membrane, diffusion is going to be small nonpolar molecules. They're going to move through there really easy compared to larger polar molecules that have to go against that gradient, that have to go with the cell membrane and squeeze through the maze that is the membrane. Facilitated diffusion is what's going to help larger things. Facilitated diffusion is going to use those integral proteins as protein channels to get from one side to the other. The protein or channel proteins create hydrophilic pores in the membrane through which small charged ions can diffuse. Watch the movie here on what a protein channel is. These things are going to change shape to be gates, so they're not constantly open. They're only open when the cell wants them to be open. Osmosis is diffusion, but it's diffusion of water. So there used to be an old poster in one of my teacher's rooms when I was in high school, and it was Garfield laying on a stack of books. And it used to say, like, Garfield's learning through osmosis. No, because osmosis is only water. Garfield was learning through diffusion. The water is going to move from hypotonic, high solute concentration, to hypertonic, low solute concentration. Isotonic solutions are going to move for equilibrium until it's equally balanced. So if you look at this picture, this is a hypotonic solution. There's more water than solute. Over here is a hypertonic solution, more solute than water. And over here, we've now balanced it. The water has moved. The molecules have not. The water has moved to balance it out. This is now isotonic. Again, in pictorial form, if you look at red blood cells, the reason why, if you were to go to the hospital and they give you liquids, they're going to give you a salt solution, a saline solution, because your blood has dissolved solids in it. And if we give you pure water, then your cells are going to blow up like balloons, because that pure water puts them into a hypotonic solution. Isotonics, what we're looking for, we want this number of solutes in the solution they're giving you to be equal to the number of solutes in the blood, and your blood cells are going to stay the same shape. Hypertonic is if we were to like give you pure salt water. Active transport is requiring energy. You're going against the gradient. The way I always describe active transport is like a clown car. So if I put a Mini Cooper outside and I've got 28 of you, the first couple I put in the Mini Cooper, no problem. But by like the 15th person I'm trying to shove into the Mini Cooper, I'm going to have to use energy to get them in there, right? I'm going to have to use all of my muscles to shove them in. That's active transport. It's going against the gradient. It's going from low concentration where everyone's spread out to high concentration where they're all jammed in together. Make sure you watch the movie on active transport. If things are really, really big, we're going to use endocytosis to bring them in and exocytosis to let things exit. This is basically a vesicle that is going to merge with the cellular membrane. As far as exocytosis, when it gets up to the cellular membrane, it basically merges. It's got the same materials on that vesicle as the cell membrane, and then it opens up and your cell gets bigger, and all of the contents of that vesicle now go outside the cell. Endocytosis, same thing. You have a vesicle coming. It hits the cell membrane, merges with that cell membrane because it's got the same components, and then opens up and everything spills into the cell. Your cell is constantly getting bigger and smaller with endocytosis and exocytosis. Both endocytosis and exocytosis require energy. This has vesicles as a means of transport. It's important to note that the structure of the cellular membrane is not static. It is constantly getting bigger and smaller with the movement of the proteins within the membrane, with the endocytosis and exocytosis. Essentially, you need to know that the structure of the Golgi apparatus, nuclear envelope, endoplasmic reticulum, is the same as the cellular membrane. Because they are all the same, it allows this transport of materials in and out using these vesicles. The RER stands for rough endoplasmic reticulum. It's how IB abbreviates it. And the Golgi apparatus produce and package proteins for export out of the cell, and they package them in these vesicles. So the product is wrapped in a section of membrane from the organelle, and that section of membrane is then fused with the cellular membrane, and the product's put outside the cell through exocytosis. This comes up as an essay question on paper 2 quite often. So the process for producing and exporting proteins. The nucleus contains the chromosomes which code for the protein through transcription. Then it goes out to the rough endoplasmic reticulum where translation occurs on the ribosomes, which produces the proteins. The proteins go into the lumen of the rough endoplasmic reticulum and are surrounded by the membrane. That membrane-bound protein is then processed at the Golgi apparatus, and then that vesicle leaves the Golgi and goes to the cellular membrane for exocytosis. Here it is in pictorial form. And that's it for today. So make sure that you know the difference between active and passive transport. Make sure you know the difference between endocytosis and exocytosis. Make sure you can label and draw the cellular membrane, and make sure that you know how proteins are produced and packaged for export.